So there's this quote from the biologist Sidney Brenner. It's honestly just been echoing in my head all week as we prep for this deep dive.
Oh, yeah. Is it the one about cells living and dying?
Right, exactly.
Yeah.
He said, you know, our bodies are made of cells that live and just as surely of cells that must die, which is it's a really profound way to look at biology.
It really is, because normally we spend like all our time thinking about how to keep our cells alive.
Totally. But when a cell just flat out refuses to die, when it ignores every single biological signal telling it to shut down,
Yeah.
that is when we get cancer.
Exactly. And that brings us to the mission for today. We're looking at this incredible breakthrough framework from the journal Cell.
Yeah, and it shows how scientists aren't just trying to, poison tumors with chemo anymore. They're figuring out how to hack the cell's own self-destruct codes to blow them up from the inside.
Which is a complete paradigm shift in oncology. I mean, for a very long time, this failure of cells to die when they were supposed to, it was recognized as the foundational problem of cancer. If you rewind to the year 2000, there was this landmark paper by Hanahan and Weinberg, and they established the original hallmarks of cancer.
Right, the things that make a cancer cell a cancer cell.
Exactly. And high on that list was something they called the evasion of apoptosis. Back then, apoptosis, which is a very specific programmed type of cell death, it was basically the only game in town.
So the assumption in the scientific community was, either the cell flips the apoptosis switch and dies gracefully, or it jams the switch, survives, and becomes a tumor.
Right. They thought of cellular death as a single binary on-off switch. But based on this review we're looking at today, biology is infinitely more complicated than a single switch.
It's more complicated, yeah.
What the authors of the Cell review make brilliantly clear is that we can't view cell death as just one lonely pathway anymore. Scientists have uncovered an entire spectrum of regulated cell death modalities.
So instead of a single light switch, it is much more like a biological Swiss Army knife.
I love that analogy, yes. There are multiple distinct blades, multiple tools, and entirely different sets of instructions a cell can receive to terminate itself.
Okay, to understand how researchers are pulling out these new blades, I feel like we have to start by looking at why the original tool, apoptosis, keeps failing us in the clinic.
That's a great place to start.
So if apoptosis is the default self-destruct sequence,
Yeah.
how does it actually work when a normal cell gets the signal?
Well, in normal physiology, your body uses apoptosis to clear out damaged cells or, cells it simply no longer needs. And it does this with incredible stealth.
Like a covert operation.
Exactly. It's highly organized. The cell actually starts to physically shrink. Its genetic material condenses into these tight little packages, and the outer cell membrane starts bubbling in a process scientists call blibbing.
Blibbing. I don't know why, but that sounds almost gentle.
It is gentle, yeah. And the final step is a process called ferroptosis. Specialized cleanup cells, known as phagocytes, they arrive on the scene and quietly consume those shrink-wrapped remains.
Oh, wow. So they just eat the dead cell.
They do. And because this entire tear-around is so tidy and contained, it doesn't trigger any inflammatory response in the surrounding tissue.
It's basically the cellular equivalent of dismantling a condemned building brick by brick, right? And then just quietly carting the debris away in the middle of the night.
That's spot on. The neighbors never even wake up.
Which makes total sense for normal maintenance. You definitely don't want your immune system launching a full-scale inflammatory war every time, like a skin cell needs to be replaced.
Right, that would be a disaster.
But how does the cell actually know when to start packing up the bricks?
So there are two main biological pathways that converge to trigger this quiet demolition. First, you have the intrinsic pathway, which is driven from the inside by the mitochondria.
The cell's power plants.
Exactly. When a cell experiences massive internal stress, say severe DNA damage from a round of chemotherapy, it activates these pore-forming proteins called BAX and BAAK.
Pore-forming. So they are physically punching holes in the cell's power plants.
That's it. They punch holes in the mitochondria, which releases internal factors that essentially act as molecular executioners.
Okay, so that's the inside job. What's the second route?
The second route is the extrinsic pathway, and that's driven by external signals. There are actual death receptors protruding from the surface of the cell.
Death receptors? That sounds intense.
They even have intimidating names like FAS and TRAIL. When specific molecules circulating in the body bind to these receptors, it triggers an internal cascade that leads to that exact same orderly demolition.
But cancer cells are master escape artists, right?
Yeah.
In the sources, they spend a lot of time talking about anti-apoptotic proteins.
Yes, specifically the BCL2 family.
Right. And the way it's described, it sounds like the cancer cell is building this massive brick wall of BCL2 proteins to block those internal executioners. So the self-destruct signal is sent, but it just hits a wall.
Let's adjust that analogy slightly because it's a bit more dynamic than a static wall. It's more like the BCL2 proteins are molecular handcuffs. Yeah. When the BX and BEG executioners are activated and trying to do their job, the cancer cell produces massive amounts of BCL2 to physically bind them up. It handcuffs them so they can't punch those holes in the mitochondria.
Oh, I see. So the executioners are there, but they are just completely restrained.
Exactly.
So then the obvious medical solution would be to find a key for those handcuffs. If the BCL2 proteins are restraining the executioners, we just need a drug that unlocks them.
And that is exactly what scientists spent decades trying to build. It resulted in one of the great success stories of modern targeted therapy.
Really? What did they make?
They developed a class of drugs called BH3 mimetics. The most famous one is a BCL2 inhibitor called venetoclax.
Okay, and how does venetoclax work?
It was structurally engineered to mimic a specific protein shape. This allows it to sneak in, bind to the BCL2 handcuffs, and literally spring them open.
Wow.
Yeah, the trapped executioners are set free, and the cancer cell immediately undergoes apoptosis.
That sounds like a total cure. So, why aren't we giving venetoclax to every cancer patient on Earth?
Well, because cancer is frustratingly adaptable, and the environment matters a lot. Venetoclax has been remarkably successful for certain liquid tumors.
You mean blood cancers?
Yes, like chronic lymphocytic leukemia, or CLL, and acute myeloid leukemia. In the bloodstream, cancer cells literally melt away when you pick those BCL2 locks.
But I'm guessing it doesn't work as well everywhere else.
Unfortunately, no. The drug has struggled massively against solid epithelial tumors, like breast or lung cancer, and sarcomas.
Oh, wait, if the lock pick works in the blood, why does it fail on a lung tumor? A cancer cell is a cancer cell, isn't it?
Not quite. Solid tumors exist in a very complex 3D microenvironment. They don't just rely on BCL2 to survive, they build vast redundancies.
Oh, so they have backups.
Exactly. If you pick the BCL2 lock, a solid tumor just upregulates a different anti-apoptotic protein, like MCL1, to forge a brand new set of handcuffs.
Right.
Or they just mutate the pathway entirely. The solid tumor just adapts.
Which brings us right back to the Swiss Army knife. If apoptosis is taking out the cellular trash quietly, and solid tumors easily adapt to that quiet teardown,
Right.
then maybe the quiet approach is just the wrong strategy for a solid mass. If you're listening to this and wondering, why not just blow the tumor up instead of dismantling it? Well, that seems to be exactly where the science is heading.
That is the pivotal realization driving the field right now. We are moving beyond the quiet demise of apoptosis into what scientists call a lytic cell death.
Lytic, meaning to burst.
Yes. We need a louder, messier death.
The review paper details two of these explosive pathways, pyroptosis and necroptosis. And these are fascinating to me because they aren't just ways for a cell to die. They act as actual biological fire alarms.
They really do. Let's look at pyroptosis first. The name itself is a massive clue to its function. It comes from the Greek pyro, meaning fire, and phyrotosis, meaning falling.
A fiery falling. Very dramatic.
It is. This mechanism is driven by a family of proteins called gastromins. When a cell senses extreme danger, like a severe viral infection or specific therapeutic triggers, these gastromins are activated.
And what do they physically do to the cell? Are they punching holes like those BIX proteins did to the mitochondria?
Similar concept, but on a much more catastrophic scale. The gastromins migrate to the cell's outer membrane and assemble themselves into massive physical pores.
Massive pores. So they're breaching the hull.
They literally punch holes in the outer hull of the ship, allowing water to rush in and cellular contents to just leak right out.
So instead of neatly shrink-wrapping the cell's contents like in apoptosis, pyroptosis just blows the doors off the hinges.
Exactly.
And what about necroptosis? That's the other explosive pathway mentioned.
Necroptosis is similarly explosive, but it uses an entirely different set of molecular machinery. Instead of gastromins, necroptosis builds a demolition crew out of a complex of proteins called RIPK1, RIPK3, and MLKL.
Okay, so a whole crew. When does this crew step in?
Well, when a cancer cell tries to block apoptosis, sometimes this necroptosis pathway senses the blockage and activates as a backup failsafe.
Oh, that's smart. How does that demolition crew actually tear down the cell, though?
The final step of the crew relies on that MLKL protein. Think of MLKL as a microscopic drill.
A drill.
Yeah. Once activated by the RIPK proteins, MLKL physically marches to the cell's outer skin, the plasma membrane, and starts boring holes right through it. It disrupts the integrity of the membrane until the internal pressure becomes too much.
Wow. But regardless of whether cell uses the Gastromin pumps of pyroptosis or the MLKL drills of necroptosis, both pathways end in the exact same catastrophic event, right? The sources call it PMR.
Plasma membrane rupture. It is the ultimate cataclysmic final event for a dying cell. And what is truly incredible is that scientists recently discovered the specific protein that drives this final explosion.
Which protein is that?
It is called NINJ1. For decades, we just assumed that when a cell burst, it was a passive process, like, a balloon popping because it got too full of water.
Right, like if you drill enough holes in the membrane, eventually it just gives way naturally.
That's what we thought. But the discovery of NINJ1 proves that membrane rupture is actually an active, genetically encoded process.
Wait, really?
Yeah. The cell purposefully polymerizes this NINJ1 protein to actively shred its own membrane to pieces. It is a deliberate suicidal explosion.
Okay, so the cell actively blows itself to pieces. Why does this matter so much for treating a patient's tumor? Why is this violent, messy death superior to the clean, quiet death of apoptosis we talked about earlier?
Because that messy spillage solves the single hardest problem in cancer therapy: immune evasion.
Ah, right. Solid tumors are practically invisible to the body's immune system.
Exactly. But when a cell actively shreds itself via pyroptosis or necroptosis, it forcefully ejects its entire internal contents into the surrounding tissue.
And what is in that spillage? The paper keeps mentioning DAMPs, damage-associated molecular patterns.
DAMPs are the key. Think of DAMPs as biological shrapnel.
Shrapnel, I like that.
These are molecules like ATP or specific nuclear proteins that belong strictly inside a healthy cell. They should never be floating freely in the extracellular space.
So when the immune system detects this shrapnel, it reacts.
Oh, it reacts strongly. It treats it like a blaring siren indicating massive tissue damage. And along with DAMPs, the bursting cell releases highly inflammatory cytokines.
So you are basically flooding the tumor microenvironment with chemical flares.
Precisely. You trigger a phenomenon called immunogenic cell death, or ICD. You are finally waking up the body's dormant immune system.
And who responds to the flares?
Specialized scouts called dendritic cells. They rush to the scene of the explosion, and they scoop up the debris, which now includes unique mutated proteins from the tumor itself.
Oh, I see where this is going. The dendritic cells are taking pieces of the destroyed cancer cell and presenting them to the immune system's heavy hitters.
Yes. They take those tumor proteins back to the T cells, effectively saying, this is what the enemy looks like. Here's their uniform. Go hunt down any remaining cell wearing this.
Wow. By triggering these explosive deaths, you are forcing the tumor to become its own personalized vaccine.
Exactly. You don't just kill the tumor in the moment, you generate long-lasting immune memory to ensure it never comes back.
That is the holy grail. But okay, let me challenge this for a second, because earlier we established that cancer is endlessly adaptable.
True.
What if a solid tumor has mutated so heavily that it is genetically disabled the BCL2 apoptosis triggers and snipped the wires to the Gastromin pores and dismantled the necroptosis drills?
It happens.
What do you do when a cancer cell has literally deleted its own genetic self-destruct codes?
That is a very real scenario in advanced treatment-resistant cancers. And the answer is, well, when a cell has dismantled all of its genetic self-destruct switches, you stop looking for a genetic trigger. You attack their supply lines. You sabotage their metabolism.
Which introduces the weirdest, most fascinating blade on this Swiss Army knife, which is ferroptosis. I mean, even the name sounds like something out of science fiction.
It does. And it is an entirely distinct phenomenon from everything we've discussed so far. Ferroptosis is an iron-dependent form of cell death driven by extreme lipid peroxidation.
Okay, let's break that down. Lipid peroxidation. Are we talking about the fats inside the cell?
Yes, specifically the lipids that make up the cell membrane itself. Under certain conditions, those lipids start to intensely oxidize. It is conceptually very similar to rusting.
Rusting.
Yes. The fats in the membrane literally rust until the structural integrity of the cell just collapses and falls apart.
Rusting cell membranes. That is wild. And the review points out this pathway was discovered somewhat by accident, right? Like they weren't looking for a new death pathway. They were screening Burkitt lymphoma cells for nutrient dependencies.
That's the beauty of basic science. Researchers were simply looking at what happens when you deprive cancer cells of specific amino acids, and they observed that the cancer cells died, but not by apoptosis.
Right. They die through this massive oxidative collapse.
Exactly. It turns out ferroptosis happens when the cell's lipid, iron, and redox metabolism goes completely haywire.
So why doesn't every cell in our body just rust away?
Because under normal conditions, cells are protected by a very specific, dedicated anti-rust enzyme called GPX4.
Okay, so GPX4 is like a biological rust proofing.
Think of it more like a cooling system. It's
Yeah.
constantly patrolling the cell and neutralizing those toxic lipid peroxides before they can accumulate.
Okay, but here's my question. If ferroptosis doesn't have a genetic on switch like the other pathways, why is cancer so uniquely vulnerable to it? I mean, why does disabling this GPX4 cooling system kill a cancer cell, but leave a normal, healthy cell relatively unharmed?
It comes down to metabolic greed and stress. Cancer cells grow at an unnatural, furious pace. To sustain that rampant division, they require massive metabolic resources.
They're always hungry.
They hoard iron, they synthesize huge amounts of lipids to build new membranes for new cells, and as a byproduct of all this frantic activity, they generate incredible amounts of oxidative stress.
So a normal cell is like a reliable sedan driving the speed limit, while a cancer cell is a high-performance Formula One car just running its engine constantly in the red.
That is a brilliant way to picture it. Because the cancer cell is running so incredibly hot, it becomes entirely addicted to its GPX4 cooling system.
So what happens if you turn off the cooling system?
Well, if you introduce a drug that inhibits GPX4, the reliable sedan might get a little warm, but it survives. The Formula One cancer cell, however, instantly overheats. It succumbs to runaway lipid peroxidation and violently rusts to death.
And this seems incredibly relevant for cancers that survive standard treatments. The sources highlight something called DTP cells.
Drug-tolerant persister cells. These are the nightmare cells that alter their metabolism to hunker down and survive traditional chemotherapy.
But there's a catch, right?
Yes. The irony is, by altering their metabolism to survive chemo, they inadvertently leave themselves uniquely exposed to oxidative sabotage. Ferroptosis exploits the very metabolic adaptations that made the cancer resistant in the first place.
Okay, let's pull all of this together. We've got the quiet, orderly teardown of apoptosis. We've got the explosive, fiery alarms of necroptosis and pyroptosis. And we have the sheer metabolic sabotage of ferroptosis.
Right, all the different blades.
Now that scientists have mapped all these different tools, how do doctors actually deploy them inside a patient?
The paper introduces a conceptual framework to guide this called the Triforce of Immunogenic Cell Death. If you really want to clear a solid tumor and ensure the immune system keeps it gone, you need 3 distinct elements to happen simultaneously in the tumor microenvironment.
The Triforce. Okay, what are the 3 pieces we need to collect?
First is antigenicity. This means the dying cancer cell must present its unique tumor-associated antigens, the targets, to the immune system.
Got it. Show them the target.
Second is adjuvanticity. The dying cell has to release those damps, the biological shrapnel we talked about, to sound the alarm.
Sound the alarm, right?
The third is inflammation. The environment must be flooded with pro-inflammatory cytokines to physically draw the immune cells into the tumor bed.
So antigenicity shows them the target, adjuvanticity sounds the alarm, and inflammation calls in the troops.
Exactly that. And different cell death modalities provide different pieces of this triforce. If you only trigger apoptosis, you might get antigenicity. The target is there, but apoptosis is too quiet. It completely lacks adjuvanticity and inflammation.
So the immune system never notices the target.
Precisely.
So what does this mean for a patient walking into a clinic, say, 5 years from now? How does their treatment change based on this Swiss Army knife approach?
It means we abandon the search for a single magical silver bullet. If you hit a cancer with only one drug that causes apoptosis, a few cells will mutate, forge new BCL2 handcuffs, and the tumor will roar back.
So what's the alternative?
The future is sequential strategic therapy.
Give me an example of how that sequence works in practice.
Okay, imagine giving a patient a round of traditional chemotherapy. This heavily stresses the cancer cells. Some die, but the strongest ones hunker down, maybe mutating to block apoptosis and upregulating proteins to suppress their necroptosis alarms.
Right, they become those persister cells.
Yes, but then you follow up immediately with a highly targeted drug that removes the brakes on that necroptosis pathway. Or perhaps a GPX4 inhibitor to trigger ferroptosis.
You force the stressed, resistant cancer cells into an explosive, fiery death while their defenses are pointed in the wrong direction.
Exactly. You are actively trapping the tumor in its own evolutionary dead end. If it mutates to escape apoptosis, you hit it with ferroptosis. If it alters its metabolism to escape ferroptosis, you trigger pyroptosis.
It's like a game of molecular chess.
It really is. The goal is to map the specific vulnerabilities of a patient's unique tumor and strategically deploy the right combination of cell death pathways to hit all 3 points of that triforce.
It is absolutely incredible. To recap the ground we've covered today, cancer's primary trait from the very beginning has been evading death. But science is finally mapping the full, complex spectrum of cellular demise.
We really are.
We are moving from relying solely on the quiet, orderly teardown of apoptosis to leveraging the explosive inflammatory fire alarms of pyroptosis and necroptosis. And when the genetics fail entirely, we are turning to the metabolic rusting of ferroptosis. We are literally learning to turn cancer's stubborn survival against itself.
And that conceptual shift just cannot be overstated. We are moving away from broadly toxic chemotherapy, which, let's face it, just poisons everything in the body and hopes the cancer dies first, toward precision cell death engineering.
Precision cell death engineering, I love that.
It is the foundation of the next 25 years of personalized oncology. The goal is no longer just to kill the cancer in the moment. The goal is to induce the right kind of death so you teach your own immune system how to keep it gone forever.
It's a hopeful vision for the future.
Yeah.
But I want to leave you, the listener, with a final thought to mull over, building on all of this. If you are listening to this deep dive and marveling at the power of these fiery, highly inflammatory cell deaths to wake up the immune system, consider the double-edged sword.
Oh, that's a really good point.
Right. If we get incredibly good at triggering necroptosis and pyroptosis to burn a tumor down, how do we stop it once the cancer is gone? If we engineer the perfect biological fire alarm to eradicate the disease, could the ultimate cure for cancer risk triggering profound autoimmune diseases where the body's fire alarm simply refuses to turn off?
It's the million-dollar question.
It really is.
Yeah.
It brings us right back to Sidney Brenner. Our bodies are made of cells that must die, but we have to make absolutely sure we are still the ones controlling the switch.